thank you so much for coming. I'm deeply honored for this award, and I'm really thrilled to come talk to you today. So I'll start out with a few caveats. For those of you in the room who are not philosophers, you might be pleased to know that I'm not going to be giving necessary and sufficient conditions on progress in science, let alone cancer science today. So I'm going to be giving some stories. I'm going to be telling some stories about myself, my experience learning about this material, and then also some stories about the history of cancer research. So my hope is that by telling these stories, I'll be able to evoke some messages, some upshots, some lessons about progress in science and how we can get stuck. And so, haha, one way in which we can get stuck is the computer goes out. Another way we can get stuck is, I think, evoked nicely by this image. So I was particularly struck by this image of these climbers ascending Mount Everest. What's interesting about this image to me is that there's these steep slopes and there is one single path. And they're inching very slowly up to the peak. And I think this nicely evokes features of certain aspects of cancer research today, which I think can be a bit stuck. And so I'm going to say a little bit about how we can get stuck and how we can do better getting unstuck. All right. So in order to say, I left off something. So some people think that a lot of current cancer science today, which is driven by multi-institutional, multi-year, multi-billion dollar or multi-billion pound investments is not making good progress and not making good on its promises. And on the other hand, some people think it's doing immensely successful work. So in order to assess this judgment, I want to try to say a little bit about what we mean by progress. That's all right. Thank you so much for talking. Valiant effort.
Failing as a mechanic here.
It's fine. I can manage. Oh, I can need to move closer to the mic.
I know it's not as older.
It's fine.
I will try and find a replacement for you. I'll be right back, but carry on.
Oh, okay. If you don't need to.
Sorry about that.
Okay, so Lakatos talks about progress, and so one of the sort of central claims he makes is, first of all, that of course we have a research program that has a series of theories. It's characterized by a hard core with a protective belt. Both a negative and positive heuristic are sort of protection of this sort of central core set of theories. And it's successful if it makes predictions and it accommodates past observations. And it's degenerating if it makes sort of ad hoc accommodations to apparent disconfirmations, if you like. And this all sounds fine and good, but of course it leaves us with a set of puzzles. How do we know how to demarcate research programs one from another? What counts as genuinely novel predictions? And when do we have good grounds to say that a research program is degenerating?
Wrong one.
I think that these are hard problems, and they are especially hard because the sort of scientific world that Lakatos confronted was quite different from our scientific world today. As I mentioned earlier, in cancer, science, and medicine, we're talking about rather a motley mix of data gathering, of analysis, of theoretical modeling, of experiment, and so there's not sort of a unified project and a unified theory, I would argue, that unifies all of these different and rather diverse questions that people are investigating. Whether you're trained in medicine, in cell and molecular biology, in genetics and genomics, or some other field, there are quite different background assumptions and standards and expectations and sets of problems that are of interest to these very different research programs. So I think it's hard to say that there's something like a unified project, and I also think that cancer itself, of course, is so heterogeneous that we should be surprised to expect that there should be some sort of unified general theory or research program guiding all of this research. So in order to sort of say a little bit about how I think I got to this point and these insights, and also to say a little bit more about why I think this is the wrong way of thinking about progress in cancer science and how we get stuck, I want to say a little bit about how and why I wrote this book. So I started this book in 2009 when I was diagnosed with breast cancer. I was very puzzled and confused. I was a young woman. I didn't know why I got cancer. And so as a typical academic would, I immediately got on PubMed and read as much as I possibly could about cancer, medicine, and science. And at the time, there was really just a handful of philosophy of science about cancer. But there was quite a bit of history, sociology of science, and also some fantastic work on cancer science and medicine and the law. And so I started here, and I think this had a huge influence in my thinking in the project of the book. And so in that way, I think Lakatos's insight about the dialectic between history and philosophy of science was very important and crucial to my understanding of how to think about the kinds of problems that are interesting and hard in cancer research. I also think it gave me a very different perspective on the nature of this research than perhaps if I had started sort of with a well-established body of research in philosophy of cancer science. Since, as I mentioned, there was just but a handful of work in philosophy of science of cancer, and most of it was focused on competing paradigms or theories. So I think that philosophers of science sort of tend to assume that what we should be looking for when we look for science, good science, better science, worse science, is thinking about novel prediction and good theories or true theories or false theories. And what I found when I was looking at this immense and helpful body of literature on the history and sociology and sort of practical import of cancer science was how much of what cancer science was doing was really problem-driven, how it was influenced by context, by social context, by economics, by a variety of stakeholders' interests. And I also realized that in order to write a book on philosophy of cancer, I actually had to learn the science and medicine behind cancer, so I took myself to medical school. And I am very, very grateful for the professors I had at the University of Utah Medical School who let me sit in on their classes, conduct interviews, sit in on lab meetings. So I spent about 3 years at University of Utah taking classes in cell and molecular biology of cancer, cancer epidemiology, and then I moved to Washington University in St. Louis. And what struck me about these two very different institutions was how different the culture of medicine and cancer science and medicine was. So at the University of Utah, most 99% of the medical students are training to be clinicians. They're not getting a PhD and an MD at the same time. At Washington University in St. Louis, the vast majority of medical students are getting an MD and a PhD simultaneously, and most of them are doing sort of the cutting edge research. Right? And in fact, a lot of the cutting edge research in cancer. So Washington University in St. Louis is one of the homes of what became one of the largest big science projects in the 20th through the 21st century so far, which was the Cancer Genome Atlas project. So once I got there, and I don't discuss this in the book, but I realized after I finished the book that I really should have, is I started interviewing all of the participants in the Cancer Genome Atlas project at Washington University in St. Louis. This is one of them here, down at the bottom, standing next to one of his sequencing devices. And I realized that there is a very different culture of science and medicine at different institutions, and that this was shaping how we think about what kinds of interesting questions, research questions, problems there are. So I want to back up a bit and say how Washington University became such a central place for doing cancer science and medicine. And how, I think, now I have to back up a bit, one of the insights I gained from reading some of the fantastic history and sociology of science I mentioned was Joan Fujimura's fantastic book, Crafting Science, in which she talks about something called the oncogene bandwagon. So I'm just going to go back a bit and point my finger at that particular book. So one of the things Fujimura does in that book is she describes how in the sort of late 20th century, advances in biotechnology made possible the close examination of these micro-factors at work in cancer, in particular what are called oncogenes. And there was this rush for cancer researchers to sort of look to the genetic level and try to discover more and more and more oncogenes, because every oncogene is potentially a Nobel Prize, or so it seemed, at least in the early stages. And I actually talked to some women scientists who told me they decided not to do cancer science because they thought it was so competitive. there was such a huge bandwagon effect on cancer science that they thought they couldn't even do children and have children and do science. So they decided to work on completely different projects because it was this huge rush bandwagon effect. So how did that happen? How did we get here? In order to sort of explain this, you need to back up a little bit to the 1989 Nobel Prize in Physiology and Medicine, which was awarded to Michael Bishop and Harold Varmus for their discovery of the cellular origin of retroviral oncogenes. Why was this so important and interesting? It was important and interesting because prior to this time, where were they looking for the causes of cancer? Sort of all over the place, but one central place was viruses. They thought maybe, perhaps, cancer was a viral disease. It was an infectious disease because there were these model organisms that showed, it seemed, that there were sort of viral causes of the production of certain kinds of tumors, in particular in chickens. The SERC gene, which they discovered, it was found was shared by all of us, and in fact, probably transmitted by a virus across species at certain points, and could lead to the onset of a cancer. So what happened subsequent to this, and especially I think there's sort of, now I want to sort of drop this phrase, prestige bias. I think there's this sort of enormous excitement around the discovery of cancer genes, but also enormous excitement around the development of certain kinds of technologies, and particularly technologies for genome sequencing. And with the completion of the first human genome in 2000 or 2001, depending on how you count, there was a lot of excitement around the possibility of looking to the genome to find the fundamental causes of many different diseases, not only cancer, of course, but the thought was, and here's Bill Clinton suggesting, that it is now conceivable that our children's children will know the term cancer only as a constellation of stars. And I think this is a fantastic image. It's striking to me that the fellow sitting next to him is Francis Collins. Francis Collins was the head of the National Institute for Human Genome Research. He was one of the sort of completers of the first human genome, and he was a big booster for genomics, and in particular cancer genomics, as a tool for not only better understanding cancer biology, but developing new drugs and treatments and interventions into cancer. So here he is. This is as of 2007, but it's shortly after the beginning of the Cancer Genome Atlas project, which was a 10-year project aimed at sequencing ultimately 33 major cancers: prostate cancer, breast cancer, glioma, a number of other cancers. And what they did is they matched tumor samples with healthy tissue from individuals who had cancer and used this information to sequence a sample of different cancer types or subtypes within each of these major cancers. So for instance, you can see at the top here, 1,088 breast cancer patients, 470 melanoma patients, and 185 pancreatic cancer patients. So one of the first questions I had for the gentlemen and ladies I interviewed who were involved in the Cancer Genome Atlas project was, why this variable sample size? After all, if what we want to understand is what are the representative genes or driver genes associated with this cancer type or subtype, shouldn't we get a significant sample, and shouldn't we get the same samples across these different cancers? And it turned out that after all, they couldn't do that, in part because quality of samples was a big problem, and there are some cancers that are exceptionally rare. And so there were problems along the way in terms of getting the samples they wanted, in terms of doing the analysis they wanted to do. And the major director, one of the program directors for the program, Carolyn Hunter, I interviewed, she ran it for the last 5 years at the National Human Genome Research Institute, said it was kind of like, and I love this quote because anyone who loves Neurath will love this quote, she was like, it was kind of like rebuilding a plane as we were flying it. Because they didn't know what they were doing at the beginning. They didn't know what to look for, they didn't know how many genes to sample, and so it was a very idealistic project, right? They were sort of learning to do as they did it. They didn't know what they didn't know. And so this is Collins promising that if we do this project, we'll identify all the genetic alterations in different forms of cancer so that gene changes driving the disease can be targeted directly. As each new type of cancer is studied and added to the TCGA, investigators will gain another rich set of genomic targets and profiles that can be used to develop more tailored therapies. So what's going on during this project? It's multiple institutions, it's billions of dollars. It's institutions put into competition with one another to sequence faster, more efficiently, and to analyze those sequences in ways that give us useful information for developing targeted therapies. So I've mentioned this already. If you haven't run across genome sequencing before, what they're doing is they're gathering these reference samples, what they get called. They're sequencing them. They're doing what gets called mutation calling, comparing the reference and the tumor, so the healthy tissue and the tumor tissue, and then analyzing them and publishing them in these marker papers. So the marker papers are the prostate cancer genome, the breast cancer genome, et cetera, and these are published in Nature and Cell over the course of that 10 years. What's interesting about this is now everybody's going back and saying we need to redo it over again. We didn't sequence deep enough. We didn't sequence enough people. So this comes from Carolyn Hutter's sort of presentation at the sort of celebratory end of the Cancer Genome Atlas Project, which then became the Pan Cancer Atlas Project, which I'll mention in a moment, in which she sort of summarized all the things they aimed to do and celebrated their successes. Right? They've made great progress. So what they've done is they've developed better taxonomies of cancer. They're hoping thereby to improve risk stratification. They're identifying what they call drivers or eventually actionable genes, which it turns out is a very interesting concept in flux that means lots of different things to different people, that are enabling development of drugs and precision therapies. They're improving technology, and they're improving understanding of cancer biology. There's what she said in public. Here's what she said in private to me in an interview. I said, can you point to one example of a really interesting advance in cancer medicine that has resulted from the Cancer Genome Project, the Atlas Project? And she laughed at me. And then I said, can you tell me on the basis of this project how many types of cancer there are? And then she laughed harder at me. And so what I got from this is roughly that she said, look, what we did was, this is when the sort of airplane and flight comment came on, is we were sort of figuring out as we were going along. It was really hard. It was enormously expensive and difficult to sequence at the beginning. We got it faster, we got it more efficient, but we didn't really know how many samples to take, which ones, et cetera, et cetera. What we did is we put all these different teams in competition with one another in service of trying to get better, faster, more information, more useful information about cancer. And you can see Washington University in the center is one of the sort of hosts of major sequencing centers. I'm sorry. So this became one of the locations for this development of a community of researchers who are very good at taking samples, developing sequences, and analyzing sequences. And I went to interview Li Ding, who is one of the cancer researchers at Washington University, and her lab didn't look like what you might think a cancer biology lab looks like. It was a bank of computers. And she had, over 100 postdocs analyzing sequence. Oh, thank you. Okay. Okay. So what struck me when I got to WashU, thank you so much, was how different the culture of science and medicine is, the kinds of questions people were asking, and the sort of culture of sort of competition that was going on at WashU. In contrast, at Utah, there were relatively few researchers involved in such big science, big data projects, and most of the medical school instructors I had were clinicians, and they were teaching about clinical medicine and clinical practice. So the culture of the institutions and the very striking difference between them made me think really hard about what it is to do good medicine and good cancer science. What counts as progress? What are the questions we should think are really interesting and important? So like I said, there's been a difference then between what the public representation of the Cancer Genome Atlas Project was for and what in private the researchers will admit to you. I think they'll admit this, which is really we were building partnerships with biotech. We were making the tech faster. We were making it more efficient. We were building communities of scientists who could analyze this kind of data. And we were trying to learn what kind of questions to ask. We didn't even know how many samples to take because we had no idea how massively heterogeneous cancer in fact was. Okay. They did make progress in one thing. So one thing that is shown on almost every public talk at the National Human Genome Research Institute is this slide. Because they are very proud of the fact, and this is one of the things Carolyn Hutter pointed me to, that it is so fast and efficient now and cheap to produce a genome. To sequence a genome, it is very, very cheap and fast. And notice, here is the beginning of the Cancer Genome Atlas project. Here's the end. Okay, that's no accident. So is this progress? Shortly after the completion of the Cancer Genome Atlas project, Francis Collins sat down with our then President of the United States, Mr. Obama, and he launched the Precision Medicine Initiative. This is where he announced that he was going to devote $70 million to, as he said, ramping up cancer genomics. And he made the exact same promises, and oh, I should say, he said this, I have to tell you this joke. Okay, ready? One of the few times in which both sides of the aisle stood up and applauded mightily, I think it was the only time, during this State of the Union address, was when he said this was going to enable them to give the right drug at the right time to every patient. Everybody said, right? But this was at the end of the Cancer Genome Atlas project. So why? Because the project opened up more questions than it answered. Here's what I mean. Why was he giving $70 million when they just finished? Didn't they finish sequencing all the major cancer genomes? So what happened as a result of this project was they found too much information. The more cancers they sequenced, the more genes they found that seemed to be drivers of cancer. And you'd think a larger sample size would help you identify the few key genes, right? But exactly the opposite seemed to be the case. The more they sequenced, they found not astronomical, but according to Steffens et al., genomical amounts of data. So this is coming out of what's called the Pan-Cancer Atlas Project, which is downstream of the Cancer Genome Atlas Project, which is aimed at trying to sort and make sense out of all this extremely complicated data and try to find common patterns and processes across very different cancers. So what they report in this paper, interestingly, is that they found greater than 3,400 putative driver mutations associated with cancers. Though this is only 299 in this image. So driver genes are genes that are taken to play some important causal role in cancer. But what counts as a driver gene, it turns out, is a concept in flux, because how we measure it and how we define it has evolved significantly in the past 10 or 15 years with advances in new technology and advances in more and more different algorithms for analyzing cancer genomes. But what it ultimately means, the sort of simple version is, does it rise above this level of frequency in a significant body of cancers? So it occurs at a certain level of frequency. But of course, everyone here knows correlation is not causation. Occurring at a high level of frequency doesn't necessarily tell you what kind of causal role these genes play, nor does it tell you necessarily whether we're going to be able to target them with drugs. So I think that this case raises some really interesting questions about progress in science. On the one hand, it yielded all sorts of hard theoretical and practical problems. Which variants are really the causal drivers of cancer? How do we know? How can we improve mutation calling, analysis, and so on. But there are also applied problems, which are, once we say which of these are the driver genes, which among them are actionable? Which, it turns out, means different things if you're a clinician versus a pharmaceutical researcher, but more or less, the idea is something like, can we develop applications, whether prognostic, diagnostic, or treatment applications using this genetic information? So as you can imagine, there's arguments on both sides. Some people say it's a huge waste of time and money. Some people say it's done more to help us understand cancer than anything else. Where's the truth? I think it's somewhere in between. And here's what I'm going to say about it. I want to briefly say something about these critics' concerns because I think they are really very important. So I recommend this fantastic book by Vinay Prasad called Malignant, How Bad Policy and Bad Evidence Harm People with Cancer. It's really more looking at the downstream end of translation than the upstream end. But one of the things he points out is that there's this enormous hype around precision or personalized cancer medicine. But the vast majority of cancer patients today are still treated with standard radiation, chemotherapy, and surgery. A relatively few proportion benefit from some of these new targeted therapies. They are enormously exciting, but they're not a sure thing. So when the Cancer Genome Atlas project was proposed, Collins pointed to magic bullets like Gleevec or imatinib as vivid examples of what we could do with the right kind of molecular information. Everyone knows chemotherapy, radiation, surgery are incredibly painful, awful things, and they cause lots of horrible side effects. So the hope of these targeted therapies is that you won't have these horrible side effects associated. And Gleevec or imatinib is one of the vivid cases of success in that case. But it turns out that a lot of these precision therapies that have been developed are not curative. So these are Kaplan-Meier plots. You don't need to know all the details, just to know that recurrence happens. So if you treat someone with these new drugs, cancers evolve. They evolve resistance, they evolve ways of getting around this treatment. And so you might have maybe 5 or 6 more months of life, which is actually fantastic for cancer drugs because we've like sort of picked the low-hanging fruit and now we're looking at like 3-5 months is really, really great. But cancers recur. And I'm going to sort of give you a warning about the next slide. So if people are really disturbed by images of cancer, please look away. Recurrence is common even in many forms of precision care. This is a RAF inhibitor in melanoma, and the patient, what's striking to me is they often show the first 2 images in cancer medicine textbooks, and like the textbooks I used in medical school, but they don't show the 3rd image. So recurrence happens even in precision care. There are exceptions, there are exceptional responders, and they are subjects of important study, but this is actually not that common. So recurrence does happen even in precision therapies. So what I guess I'm talking about here has been given a name by epidemiologists. It's called the Valley of Death. That is, understanding basic biology of cancer doesn't necessarily tell us always how to treat it. And many treatments have been developed for many diseases when we don't understand the mechanisms. And moreover, it's enormously expensive and time-consuming to develop, to get from the basic science down to effective cancer drugs. So where does this leave us? I wanted to return to this image of the climbers climbing Mount Everest in a long, thin line inching toward the top. And mention sort of one of the graduate student MD PhDs I spoke to who was kind enough to let me ask him really naive questions at WashU. And he said, I don't want to look at that. I don't want to work in that lab because they publish too much. And I said, what do you mean? And he said, well, you know, they publish the smallest publishable unit. That's the goal. And I think that this sort of points to the kind of problem of getting stuck that I'm talking about. Our current scientific funding paradigm and scientific training encourages specialization. Young scientists are encouraged to focus on very specific, well-defined problems, and as a result, know an enormous amount about very little. They're making incremental changes along a well-defined, well-marked path. And this model for science has benefits, even with its costs. A scientist choosing such a path has the advantage of joining a large network of scholars, working on the same problems, and they have clearly defined solutions. There are well-governed standards of evidence and specific publications and journals that they know what to do to publish in. They know the rules of the game. However, I think this model of scientific research can lead to a very narrow conception of the interesting problems worth exploring. So I want to suggest that critics of the oncogene paradigm who are still sort of raising their fists, and critics of precision medicine like Vinaya Prasad, their concern is not with false or bad theories. Their concern is not with bad science. It's with stuck science. It's with the values that are driving the choices about what we invest more time and money in. And so I want to rethink a vision of cancer science that looks a bit different. I want to suggest that some of the problems of silos be eliminated by encouraging more interdisciplinarity, maybe even having scientists take classes in history and philosophy of science and medicine, and investigating more causes at multiple different temporal and spatial scales. But I also think that there are patient-facing research questions and preventive science questions that we need to look at more closely. Most of my undergraduates at Washington University in St. Louis who are pre-meds and want to go do MD PhDs think of the future of cancer science as looking like this. There's a huge amount of prestige bias. There's a huge amount of funding, and there are jobs and there are opportunities here. But one of my favorite epidemiologists, Peto, suggested that if we really care about preventing cancer, we need to take a black box instead of a mechanistic research strategy. What does he mean? Oops. He points out that almost all the most important knowledge we have of how to prevent cancer comes from black box epidemiology. So pointing to the example of Dahl and Hill in 1950, their publication looking at the link between smoking and cancer has done arguably more to prevent cancer than any molecular signatures, diagnostic tools, or what have you. So our choices about what research to pursue are value-laden choices. They are shaped by political, economic, social interests, the organization of institutions, various forms of inertia, not only on where the science is leading us. And what we choose to study and what we consider good science is informed by these bandwagon effects and entrenched practices and models of success. But we could think about different kinds of future of research cancer science, looking at, for instance, smoking cessation programs, studying disparity in incidence and mortality, environmental carcinogens, psychological care for patients and families, addressing symptoms of treatment, recurrence, palliative care, and ultimately end of life. One of the least well-funded areas is end-of-life decision-making. And I think that's a huge problem, given that so many of these hard questions that patients are facing when they're going desperately to those precision medicines are, I've tried everything else, right? Where do I go now? And sort of the process of deciding to accept palliative care is an enormously difficult process and requires a lot of support and better support than we have today. Thank you so much.